Hi, this is Pinky Devi, PhD student in Dr. Carol Miles' Vegetable Horticulture program at
This
Washington
video
State
is about
University.
one of our vegetable grafting research study that is optimizing watermelon grafting to enhance grafting efficiency.
Grafting has become a common practice for watermelon production worldwide to mitigate against biotic stresses like soil-borne diseases and abiotic stresses like temperature extreme, soil salinity, etc. Verticillium wilt is an example of soil-borne disease that decreases the production of watermelon all over the world. This disease is caused by the fungi called Verticillium dialii that can persist in the soil for up to 14 years, and it has over 400 host species. In case of watermelon, there is no genetic resistance and also there is no effective chemical control method. These two pictures are from a field which is naturally infested with Verticillium dialii, where you can see almost dead non-grafted watermelon plant, and there is a very healthy grafted watermelon plant. And these two pictures were taken from the same field on the same day. Therefore, to take advantage of the grafting, in North America, the most commonly used grafting method is one cotyledon grafting method. In this method, first we cut the rootstock at about 60° angle with one cotyledon remaining on the plant. The angle cut should also remove the apical meristep in the remaining cotyledon. Then we cut the scion at about 60° angle below the cotyledon where its diameter matches that of the rootstock. Then we join the two cut stems together with a commercially available grafting clip. For additional support, we can put a plastic straw. However, the watermelon grafting technique is not widely adopted in the United States due to two reasons. One, high labor costs. Second, the cost of the grafted plants. One of the major concerns related to this problem is rootstock regrowth. If we do not remove the meristematic tissue lies below at the base of the remaining cotyledon, that eventually grow as regrowth and compete with the scion for water and nutrient and eventually death of the grafted plants. To avoid this issue, we need to scout the plants in the greenhouse and as well as in the field, which is very time consuming and labor intensive method. Therefore, our study objective was to develop splice grafting technique for watermelon where both the cotyledons are removed to increase grafting efficiency and reduce the cost of the grafted plants. However, the major concern with the splice grafting technique for watermelon is the low survival rate. Therefore, we tested the external application of sucrose in combination with commercially available anti-transparent solution applied to the rootstock seedling before grafting to see if it can increase the survival of the splice grafted watermelon plants. In this method, after rootstocks are treated with the treatments, we first cut the rootstock at about 60° angle just below both the cotyledons. Then we cut the scion at about 60° angle below both the cotyledons. Then we join both the cut stem together with a commercially available grafting clip. Immediately after grafting, we placed our grafted plants in a healing chamber with controlled environment following a site-specific protocol. We recorded the survival of these splice grafted plants till 21 days after grafting and found that plants that received sucrose in combination with anti-transparent solution that had higher survival, which was 91%, as compared to the plants which were only treated with the water was 25%. From our study, we can conclude that drains application of sucrose solution in combination with commercial anti-transparent solution to rootstock seedlings before grafting can definitely increase the survival of splice grafted watermelon. Splice grafting technique can increase the grafting efficiency as it takes lesser time as compared to the commonly grafting techniques that used for watermelon production. Splice grafting could also potentially decrease the cost of the grafted plants as it would eliminate the extra attention that is required to eliminate the rootstock regrowth, which eventually decrease the labor cost that's involved in watermelon grafting technique. In 2019, we also evaluated the fruit maturity and quality of splice grafted and one-cotyledon grafted watermelon at Hermiston Agriculture Research and Extension Center and found that the splice grafted watermelon retained the same fruit quality as one-cotyledon grafted watermelon and non-grafted watermelon. However, more studies are required in the field setting under varying environmental conditions. If you want to know more about this whole study, you can go through our publication in Hot Science, which I have given details here.
This research study was conducted at WSU Mount Vernon Northwestern Research and Extension Center, and it was funded by United States Department of Agriculture Specialty Crop Research Initiative grant. Thank you for watching this video.